Hello there. Welcome to another episode of Catalyst, a platform for the scientific community to connect, discover, and transform. This is your host, Kedar Aris, and today it's my pleasure to connect with Dr. Dave Jones, Assistant Professor at the University of Michigan, Ann Arbor, in the Department of Pharmacology and Internal Medicine. Dave's primary area of research is in cardiac physiology and pathophysiology. In particular, his lab studies ion channels that regulate membrane excitability and seeks to identify mechanisms of cardiac arrhythmias and sudden cardiac death. His group uses electrophysiology, cell biology, and molecular biology techniques in a variety of systems, including stem cell derived cardiomyocytes, to identify triggers and treatments of cardiac diseases. Dave has an extensive publication record and has been a past recipient of multiple NIH awards, including the T32 and the prestigious K99 award. Incidentally, when I was preparing for my K99 grant application, Dave was kind enough to share his K99 application, and it proved immensely valuable, and I'm grateful for that. So it's my privilege to welcome Dave to the show. Good morning, Dave. Welcome to the Catalyst podcast.
Thanks, Kedar. It's nice to be here.
To begin with, for the benefit of our audience, can you briefly walk us through your academic transitions from PhD to postdoc to eventually landing as an assistant professor?
Sure. So I started out, I did a PhD at a primarily undergraduate institution in Canada called Simon Fraser University. There I was looking at the cardiac sodium channel, NAV 1.5. I was working with Peter Rubin, who's worked for a number of years in sodium channel biophysics. And then I had a co-PI as well, Tom Clayton, and the two of those helped see me through my studies there. So as a PhD, I was studying primarily how protons or acidosis modulate sodium channel behavior, with the idea that that is a contributing factor in sudden cardiac death during ischemia. So that took me about 5 years or so. And that was a really good experience. Working with Peter and Tom really showed me how to love science at a very fundamental way, and to be excited to come into the lab every day. So that was really fun. And, I had great chemistry with my PIs, and, we published a lot of work during that time. And then towards the end of my PhD, I started searching around for a postdoc. And I was looking for a lab that, I was looking for a certain intensity. Peter's lab is a phenomenal place to learn to love the science, and Peter's approach to science kind of goes along with that idea of sort of a laid back attitude, but not in a bad way. I mean, Peter has a remarkable way of inspiring his students and trainees to work their best. And it was an absolutely phenomenal experience. Going into my postdoc, I was looking for someone who would perhaps push me a little harder, because I wanted to see, if I had that motivator to push beyond what I was comfortable with, what I would be able to achieve, I guess. Mm-hmm. So that's how I settled with Gail Robertson. And that was, again, a really good working relationship between the two of us. We sort of had a yin and yang type of relationship where our skill sets were very complementary to each other. Gail has a very strong sense of science and passion for that. And she's very good at asking lots and lots of questions, and that really pushed me to understand the things that I was studying a lot further. So with Gail, we worked a lot with the hERG potassium channel, which is really what she's known for. She's done a lot of really groundbreaking science in that field. And so there I was looking primarily at how subunit abundance of the hERG potassium channel modifies cardiac electrophysiology. I learned a lot of tools while I was there. That's where I picked up stem cell science and started looking at differentiated cardiomyocytes as a model of the human heart. And so again, I was with Gail for about 5.5 years, and we published a number of really exciting papers, and I really enjoyed my time there. The University of Wisconsin at Madison is a beautiful campus with a great place to start a family, and it was a unique place because it has this cheaper cost of living, so you can easily manage your life on a postdoctoral salary, and have young kids and still be working a lot of hours with postdoc as that goes. But that was another really good experience, and yeah, so about 1.5 years out before I was done, I got the K award and started the interview process. And that ultimately led me to Michigan, which I can't imagine a better fit for me right now with where my science is and where it's going.
It's interesting you mentioned your transition in terms of culture from PhD to postdoc. And I had something similar, perhaps, because I did my PhD at Utah under Rob McLeod. The funny thing is he's a Canadian too. So there's this thing about Canadians, they're such wonderful people, and he's been an amazing mentor. But my PhD experience, and I loved every minute of it, was just laid back. There wasn't like you got to do this, you got to do this, you got to do this. It was like, you need to be self-motivated and just manage your own pace. And I had a wonderful experience with my PhD. And when I transitioned into Dr. FML's lab, that was a complete different cultural experience where it's like almost like being on the East Coast. Everyone's in the rush and everyone's like pushing, and it forced me out of my comfort zone. But I think it helped immensely. Like you mentioned, that it forces you to move out of your comfort zone to pick up new things and new skills. And I'm immensely grateful for that as well. So I think I can relate a little bit in terms of being on both sides. And there's a place for that, I think, for both.
Yeah, definitely. I try to incorporate both of those styles with my trainees, in the context of when is it time to kind of sit back and let them drive themselves, and explore a little bit on their own, and when is it time to kind of put the gas pedal on and encourage them to elevate a little bit in a different way. So I think those have been really good experiences for me.
Now, you seem to have some amazing mentors that have helped you along the way. Your PhD mentor, you had a great relationship with. And I met Gail a couple of times, and she's such a great person to talk to. And she obviously had a great influence on you. Can you speak about the role of these mentors in your professional career in terms of directing you on a path? And as an assistant professor, do you still seek out mentors to help you along the way? Do you rely on the mentors you have already established as your master team, as a sounding board? Can you just talk a little more on your relationship with your mentors?
Yeah, no, I think I've been extremely fortunate in the people I've had essentially looking after me. as I went through my training. Both Peter Rubin and Tom Clayton were just absolutely phenomenal. I mean, and they they were so thoughtful and supportive of all the goofy, misdirected things that I was doing as a PhD student, as I was figuring out how to balance all the stuff that was going on. In grad school, I had 2 kids at the time, and they were very accommodating to letting me figure that out on my own, while also still just inspiring me and being supportive when I goofed up. And then Gail was just phenomenal, kind of in the same way. I mean, she recognized that I was balancing life at home and life at work, and she let me kind of figure out my schedule. But she was always there to kind of remind me of the importance of my postdoc. This is your shot. You you got to make it happen here. So she was there to give me the tools I needed to do what I wanted to do, and make sure that I stayed on track as well. Now, as an assistant professor, I kind of I have a better sense of all the work that goes on behind the scenes. Kind of, it's there's so much communication that goes on between professors about who's doing well, who needs more help, and those sort of things. It's hard to see when you're a postdoc and a graduate student how much additional support is going on that you don't see. Thesis committee is talking amongst each other about what the student still needs or where they're shining and those sort of things. And it's really impressive. And so when I look back at all the help they've given me, I'm really appreciative of that. And as a professor, I still reach out to Gail and Tom and Peter for guidance on things. And, I have a number of people at Michigan that I go to for feedback on everything, really. Grant writing, Ames pages. Do you think I should hire this person? Challenging situations with graduate students. I got some really honest advice from a colleague once shortly after I got my K award, and I was gearing up to make the jump, and I found myself sitting at a table with 4 or 5 tenured professors, so that informal discussion that really that's really where you can learn a lot.
Absolutely.
And these folks were just peppering me with advice, and one of them said, you get the job doing the science, you're going to go into the job, and it's likely that you'll never do that again. Everything changes once you get on the other side. And I think that was good for me, knowing that and being prepared for that and being open to learning just a whole new skill set with regards to the science, and being prepared to trust someone else to do the recordings for me, which frees up my time to write and think and interact with people in a different way.
That's a great point, and that is a perfect segue into the next set of questions I had for you. One was, you picked up your skills as part of your PhD and your postdoc, and then you set about establishing your lab, and now you get your time spent managing grad students, mentoring postdocs, being pulled into different faculty positions, teaching, and so on and so forth. When it comes time to expanding your research portfolio beyond what your current area of expertise is, do you are you able to make time to build on your areas of expertise to say maybe explore another area of expertise, pick up new skills? Or at this point you have what you have, and if there are new skills to be picked up, you got to rely on collaborations?
Um, so we we're constantly looking for new techniques to bring into the lab. I think that's sort of integral to staying fresh and being able to answer sort of cutting edge questions, is making sure that your tools and techniques are at the edge of science too. I mean, that isn't to say that there aren't questions that can be answered, and fundamental, phenomenal questions that can't be answered with age-old techniques. But incorporating cutting-edge science into that, I think, facilitates things. So, I have a lot of experience with, for example, stem cell-derived cardiomyocytes, which is a rapidly evolving field. Stem cell science is constantly changing. In our most recent R01 submission, we tried to incorporate some of the advances that have been made in that field, so that we could, provide a broader impact to the experiments that we were doing. I mean, that's one way. Those adaptations come through collaborations, and you learn new techniques from other PIs, or you bring in a new postdoc that has a different skill set, but you bring those into the lab and make that part of your culture, and that becomes something new that we're able to do. So that's one thing that we're doing. We're also trying to branch out into differentiating neurons through collaborations with people who have that expertise, but that comes back to us, and that becomes something that we're familiar with and that we can do, and that helps us widen the number of questions that we can tackle.
That's a great point. Briefly, when you were applying for different job opportunities, did having a K award make it easier to get in the door in terms of the interview process, or was it not that big a deal?
I wasn't in the room when they were sifting through applications, but I would venture a guess that it did. Just now as an assistant professor, and being on the other side of selection committees and that sort of thing, it does bolster your application. It's not the only thing that can. Michigan, within the last few years, our department at pharmacology, we hired a number of new PIs, and not all of them had K awards, but they all had aspects of their CV that made them stand out in one way or another. And a K award is just one example of that. But yeah, it'll certainly help you, I think.
Got it. And was Michigan on your short list for a particular reason, or did it just happen to be a perfect opportunity for you?
It was the departmental chair that stood out to me. She is a scientist that I had admired for a number of years. Before I started working with her potassium channels, I was a sodium channel guy. And our chair, Lori Isom, made a lot of really fundamental groundbreaking discoveries with the beta subunit of sodium channels. So she's a person that I had read a lot of her work and was familiar with. And then, we start interacting at Gordon conferences and whatnot. So when an opening came up in that department, that was something that stood out to me, a place that I would like to work. She has a great vision for science, and she's also really committed to training everyone at all levels, including, young professors. There's a lot of support here for me to navigate the system, the scientific world. So yeah, I think Lori was a big part of that. And there's other really great aspects about the university that I didn't really find out until after I interviewed. I came here and I met with the other professors, and I saw the support that the university is providing for their faculty. And I saw the tools that would be available to me here. So that is really what elevated it in the end. But I think Lori is really what initially attracted me to applying here.
Is that a website like Nature Jobs where you look for jobs, or is it usually through professional networking that you identify, oh, there are certain openings available?
I think about half of the jobs I applied for, I found out through talking to people at conferences, and then the other half were through, alerts, searching the internet and that sort of thing. So yeah, I was about 50-50 with that. I mean, I found out about the job at Michigan through Lori. It wasn't through.
And can you kind of share your challenges, if you will, the first year at Michigan, establishing your lab? Was it easy? Was it difficult? What should a new assistant professor be aware of as they go about establishing their lab?
I mean, I guess I would say get used to uncertainty. It's sort of like when you become a new parent. All of a sudden, you're making all the decisions, and you have no idea really if it's the right decision. So you just have to draw on your, the experiences that you had before, and you ask insane number of questions. But I think the truth of the matter is, that first year or two, you're just hoping that you're making the right moves, and hoping that it all works out. You have, I put a lot of trust in the people that I was talking to, that they were giving me good advice, and I think they were. I'm still here, so that's good. But the uncertainty is pretty daunting at first. And you sit down at your desk for the first time and you think, well, what do I buy first? That's kind of what you just start buying stuff, and man, it's just wild in that regard.
Got it. Did you have any help or mentoring negotiating your startup package, or typically that's a tough discussions and you just take what you get?
When I was a postdoc and I was getting ready for all this, people kept saying, in every transitioning to postdoc, from postdoc to PI status, they said, don't hesitate to negotiate your package. And I didn't negotiate a single thing with the University of Michigan. But that Lori sent this offer letter, and I read it and I was like, this is phenomenal. And and I took it to Gail and I said, hey, can you look at this? And she read it and she's like, that's how you recruit somebody. I was like, right? There was no need to negotiate. It was just so, I had everything that I needed. I saw no reason to try and counter any aspect of that. But I know that that's not the case everywhere. But when I received that offer, there was no need to negotiate. They offered me.
That's great to hear.
a phenomenal package. So I was on board.
Got it. Can you briefly share about your research and just for the benefit of the audience, the kind of research you do, and what are some of the challenges in terms of doing that kind of research, and what excites you about doing that kind of research, and what's coming down the pipe 5 years from now?
Yeah. So my lab is primarily cardiac electrophysiology, and we study a protein called hERG, which is a voltage-gated potassium channel, and it regulates membrane excitability in cardiac tissue. We use a number of models to explore that, but I think the one that everyone really likes to hear about is stem cell-derived cardiomyocytes. So we use that as a model of the human heart to identify mechanisms of arrhythmia. So most recently, we've been honing in on aspects of fetal and neonatal arrhythmia. There's this bizarre aspect of human physiology that the human heart is 6 times more likely to have sudden cardiac death in the first year of life than any other part during adolescence or childhood, which is really kind of remarkable when you think about it. I mean, infants are such a precious thing, but there's just fundamental differences about how an immature heart works that we don't really understand. And it's maybe not as surprising as you might think, because, where on earth are you going to find infant cardiac tissue? You can't get that. That's just not an ask that I think most, if any, researchers are comfortable with. Asking a parent who's just going through the biggest loss of their life, can we study your baby's heart? So we're trying to take advantage of some of the advances that have made in stem cell science. Previously, we could only mature cardiomyocytes to kind of a stage of, a fetus. But there's been a lot of steps by Todd Herron, Tim Camp, Adam Helms. A lot of groups are seeing new tools that can push that maturation to something that is starting to look very similar to, isolated human adult cardiomyocytes. I mean, they're not there yet. They're not perfect, but we do have a much broader window of that maturation process. So we're trying to take advantage of that and to identify what it is about the immature heart that predisposes it to arrhythmia. Obviously, that doesn't happen in a vacuum. It's not just the heart. There's other aspects of human physiology in the brain and throughout the body that are contributing to that. But we think that the heart is going to be a major factor there. So, our most recent R01 was focused on that, trying to identify molecular determinants in immature cardiac tissue that contribute to that elevated incidence of sudden cardiac death. So we use the stem cell-derived cardiomyocytes. We're also using some rabbit models in neonatal and adult rabbit heart. So trying to pin down mechanisms of things like intrauterine fetal death, sudden infant death syndrome, and the whatnot. So that's one thing that's coming out of our lab. And then, we look at other things. I have a graduate student right now that's just looking at neuronal differentiation, because we want to see where that might go. So hERG is also expressed in the brain.
Mm-hmm.
Our potassium channel is found in the brain. And there's been some great work done by a number of scientists looking and demonstrating that hERG isoforms, there's 3 in the brain, do modulate neuronal firing. So we're kind of curious to take that sort of from a cardiac slant. We see these cardiomyocytes with this huge incidence of arrhythmia when KCNH2 is disrupted. And now we're kind of interested to see if that's going to affect neuronal firing as well. I mean, if you have a dominant negative mutation in the heart, that's going to be a dominant mutation in the brain. So we would expect some effects on neuronal behavior. And a lot of that is sort of uncertain. We don't know how the other isoforms are going to respond to that, but we've started differentiating the neurons, and we're doing some baseline recordings to validate our model. But we think that that's going to be a really exciting new direction for the lab. I think that's a good example of how collaborations can really facilitate bringing something new, a new technique into your team. So we partnered up with Mike Euler and did some CRISPR mutagenesis to manipulate hERG, and we've also got some patient-derived lines. But I think that's going to be a really exciting new direction, and it's also going to require a lot of reading on my part to shore up my knowledge of how the brain works. I feel pretty comfortable talking about the heart, but the brain is a whole nother world of complexity that will be exciting to dive into.
Absolutely. And I think I often hear it more and more. There's such a confluence of the brain and the heart, especially when it comes to electrophysiology research that we do. There's so many common things, and it seems only natural for someone who's doing hard research to as readily and quickly adapt to finding what's exciting on the neuroscience aspect of it, especially the electrophysiology side of things. So that's pretty interesting to hear. I just wanted to wrap up in the next few minutes. And to that end, I wanted to ask you, given where you are right now, as some of your experiences and knowledge and where you are right now, looking back, are there things you would have done differently, either in terms of professional networking, in terms of maybe skills that you might have picked up in addition, or maybe focused on something else? Or are you pretty comfortable that you were fortunate enough to make all the right decisions to land up here? What kind of advice would you give to people who are listening and say, hey, this is what you should do or shouldn't do?
I can't think of anything I would have done different. I have my dream job right now. I love coming into the office. I love coming to the lab. I love the science that we're doing. It's a challenging time and kind of has been for a while. We had the recession in 2008, which had its effects on hiring in academia. And now, a lot of universities are in a hiring freeze. Michigan is one of those because of the effects of COVID. But, if going into academia is what you really want to do, then, stay focused on that. And it's probably going to happen eventually. Just make sure that you're researching something that you're interested in, so you have that sort of innate passion for it. I think that'll really help you. That's what I tried to always do. But I'm just me. That's just the way that I did it. But looking back, I don't have any regrets. There's nothing that I would have changed. Where I am is wonderful. I love it here. So,
and then you bring up a great point to have passion for the kind of work you're doing. Because one of my mentors, when I was transitioning from PhD to postdoc, I asked, should I go into academia? Should I go into the industry? Should I go someplace else? I don't know how best to use my skills that I have picked up from my PhD. And he said, you could pick whichever direction you want. At the end of the day, it's going to be hard work in order to succeed. So if you're going to do hard work regardless, find something you enjoy doing, as opposed to just doing it because everybody else is doing it. So I think it really resonates with what you're trying to say in terms of if academia is something that you want to do, find something you're passionate about and focus on it. And more likely than not, your passion will drive you to success as opposed to just trying to do what everybody else is trying to do.
Yeah.
That's a great point. Thank you, Dave, for your time. I greatly appreciate it. Thank you for sharing your insights and experiences. And with that, we'll end this episode of The Catalyst.
Yeah. Thanks, Kedar.
With that, we will wrap up this episode of the podcast. If you liked what you have heard, I encourage you to share this episode with a friend. I'd also love to hear from you and how you are striving to be a catalyst. So I invite you to leave a comment on my Twitter or website. This is your host, Kedar Aris, signing off. Thank you for listening, and until next time, connect, discover, transform.